Hello, everybody. Thank you so much to the organizers for the invitation to this workshop. These are so fun. And for the invitation to speak, I'm really excited to share this story. It's not quite extreme statistics, but I think it's somewhat related to Dennis's question in that when do biologists come and talk to mathematicians? And so I'm going to tell you a story that's extreme in maybe more the colloquial sense than in terms of extreme statistics. But so I do want to first acknowledge the collaboration team that this is a part of. We have this awesome team of mathematicians and biologists that are working on this really incredible problem of this extreme behavior seen in microtubules and neurons. And so I do want to have maybe a little more biology. I think maybe I'm doing the second part of the NITMB biology inspiring math. So just so we're on the same page, right? What are neurons? Right, neurons are highly compartmentalized cells. They have a dendritic region. They have a cell body. They have a long axon. I need to remember which pointer to use. And you can think about neurons being in two different types of systems. You have the central nervous system. Think of this as the command center. You have the brain spinal cord. You also have peripheral nervous system. Think of this as you've got sensory neurons and motor neurons that are sort of sending information to the command center. But the really interesting aspect about these cells is that CNS neurons typically do not regenerate in terms of an injury, right? Imagine you sever your axon. These cells will not regenerate. You will have a problem, whereas the neurons in the peripheral nervous system do have this regeneration property. And this is where we're going to be living today. We're going to be living in a specific type of cell. This is a Drosophila larva outlined in blue, and we're going to look at these neurons that have this highly branched topology. So go from this biorender figure to this figure here is where we'll be living in terms of domain of interest. So in terms of today's roadmap, underneath these cells, right, we have this microtubule cytoskeleton. This turns out to be really important. I'm going to highlight some biological behavior that we see in the lab. I'm going to talk to you really sort of broadly about some of the stochastic models we've built and developed to try to recapitulate the data that we see in the lab, and then really kind of go to the new part of the story. So this is very hot off the presses. I'm trying to model this extreme event that I keep alluding to, but I have yet not, told you, but well, in a second. So, right, so what are we talking about in terms of microtubules? A lot of times when you think about microtubule movement, you're thinking about centrosomal searcher. You've got microtubules searching for chromosomes during the cell division. Right, we've got this idea of these motor proteins that are walking along the microtubule array. But in this context, I want to think about microtubules as bundles that are moving within certain parts of the cell. So we're not attached to anything. We've got dynamics on both the plus and the minus end of the microtubule. And really, these cells, neurons in particular, are some of the most long-lived cells. They're also some of the longest cells in organisms. So you can imagine that having a stable structure made up of microtubules is incredibly important for healthy cells in general. But what do microtubules do? Another part is I want to also give you a sense of what kind of data we have, what kind of resolution that we're dealing with this in vivo system. Microtubules have been thought about a lot in vitro settings. And so here what I'm showing you is an in vivo movie of what the lab is doing is tagging the plus ends of these microtubules. It's called EB1B. And you can see sort of these like black comments moving around. And so that's telling you you've got microtubules moving throughout this domain. So what do microtubules do? They grow and shrink through this process called dynamic instability, where you have this GTP cap shown in red, where you've got tubulin dimers that are being added. And then there's this stochastic switch called a catastrophe, where you go from a growth phase to a shrinking phase. But the key thing I want to mention is that the Rolls lab believes that this is happening at both the plus and minus end. So you've got this dynamic instability happening at both end of the polymer. And in addition to that, we even have some data regarding these types of growth velocities and shrinking velocities too. So here on the right, we've got some distributions of minus end and plus end growth speeds. Similarly, we also have how long do these polymers spend in growth and shrinking as well. I feel like I'll come over here too to walk by this screen. And then moreover, right, I've got this video here showing you the pretty stark dynamic difference between a plus and a minus end in these microtubules, where in pink you have this slow, sustained movement away from the cell body, but then all of a sudden you see this blue arrow rushing down, meaning we've got very different dynamics at either end of the polymer. Okay. Additionally, what's really interesting about these cells is they have a very distinct type of structure in that what are the orientations of these microtubules? It's known throughout many different organisms that the axon tends to have majority plus end out microtubules, but in mammalian cells, often dendrites can be mixed. So you've got both plus end and minus end out microtubules, meaning who's pointing away from the cell body. But in this organism, we have almost all minus end out microtubules within the region of the cell. And this is stable throughout the lifetime of these neurons. This is by and large consistent throughout its life. But what can happen? What's the extreme aspect of this? The Rolls lab, now over 15 years ago, had this instance where, well, what happens if we injure the axon? What can happen? So here in this image, I'm showing you a healthy neuron. It's highly branched, right? You've got 3 dendrites. This arrow is showing you where the plus ends are pointing. So this is a healthy cell. And then what happens? Well, let's cut the axon off. You can ablate this organism. And what happens is 2 of the dendrites get this mixing property of both plus end and minus end out microtubules. And then all of a sudden, one dendrite just starts having plus end out microtubules. In addition to the microtubule content changing, you're also having this dendrite grow in length as if it's trying to replace that axon that was lost. And there's even movies of this. So I recently got this from Melissa Rolls. And so this is actually showing you how this old dendrite morphs into an axon. And you can see in green is the plus end microtubule content. And you can see it's creepily growing and trying to find where that axon was connected downstream. So, so we have this massive rearrangement in the microtubule content within these cells after severing. And what's really amazing about this system is what the mechanisms to which this happens are very unknown. The lab has been trying to tease this apart for several years. And this is part of a big project with these collaborators to try to use mathematical modeling to test what mechanisms are sufficient, what's important in this process, and how can math modeling sort of tease apart this massive rearrangement. And so the first 2 stories that I want to talk about mathematically is first, how are polarity and microtubule behavior, how are they governed in a healthy situation? And then what are some new things that we're thinking about in terms of this extreme injury event? How can what sort of mechanisms are important there, and how can math help us understand those better? Okay, so first idea, we want to describe microtubules in a mathematical context. So we're going to go from this somewhat complicated geometry. We're going to zoom in. We're going to assume that we have an ensemble of microtubules, say n many of them. They're all positioned in the plus end out on one side. So you can imagine this is just all the microtubules have this 1D growth. The minus and plus ends are all in the same direction. And we're going to have 2 mechanisms that are going to limit length. And I'll explain why we do that in the next slide. First, right, we have this protein-dependent growth. This is going to be important. Microtubules use this protein to lengthen. When they're in a shrinking phase, this protein is going to become available in the cytosol for other microtubules to use to grow. The second mechanism is going to be related to this switching end state, right? So these microtubules are switching between growth and shrinking. We can model this with a continuous time Markov chain framework where you've got growth. You can switch from growth to shrinking by some rate related to the time spent in growth. But here, this catastrophe rate, we're going to be describing it as to be length dependent, meaning the longer the microtubule becomes, the more likely it is to switch into shrinking. It's believed that there's some kinesin motors that can bind and sort of depolymerize the microtubule. If the microtubules are longer, there's more binding sites for this motor to bind. And so our question was, okay, with this framework, how do these mechanisms impact microtubule dynamics? But 2, can we recapitulate the data that I showed you before, right? The speeds that are realistic, the time spent in growth, and so on. So the first thing was, well, what happens if we have no length regulating mechanism, right? So let's just say, right, we have the time these microtubules spend in growth and shrinking. Let's just input that into the stochastic model and without any other mechanism, see what happens. And you can notice here on the left-hand side, I'm showing you a plus end of a microtubule and it's given minus end. So showing you that we've got sort of large growth in the plus end, small growth on the minus end. And then we see this average length in red increasing. Sorry, we've got window washers now. That's They just zoom down here, so they get a free talk. So what does this look like when you have both length regulating mechanisms? The main thing I want you to take away from this is, one, we are regulating length. So we can hit, maybe we have a target length we want to achieve based on what our collaborator says she thinks is inside the cell. We're able to target that. Moreover, what I want you to notice here on the left is you'll notice that the plus and minus ends, they come together, right? So the microtubules are shrinking to a length 0. We're going to assume we have a fixed number of microtubules. Once a microtubule, quote, dies, we just reseed it in the next simulation and let it grow as it wants to. So even more, another sample of a run with a different ensemble of microtubules, just to show you the dynamics of these filaments, right? You can see they're dying, regrowing, and so on. But right, what did I really show you? That we're able to limit length, but we really want to target experimental data. Yeah.
I missed something really crucial. When you have the catastrophe, does the microtubule unravel completely, or does it stop somewhere?
It will stop. So, well, when the catastrophe is going to be that switch from growth to shrinking. So it's going to be in a shrinking state, but then there'll be some type of rescue event later on. However, it can shrink to that length 0. So we would call that a death. But yeah, you can switch from a shrinking state to a growth state.
And so you just have some probability of stopping, equal probability of stopping at any point.
Yeah, right. You've got this. Yeah, exactly. So there's a lot of knobs to tune, right? There's a lot of parameters to input into the stochastic framework. We're achieving a limited length, but can we achieve our target length, and can we achieve other desired outputs that we see in the model? And the main result from this first paper that came out a couple years ago is we developed this coarse grain ODE framework. Using a steady state analysis of that framework gave us relationships between different parameters and different behaviors that we may want to achieve, right? So if we have a target steady state length, what does the amount of tubulin need to be, or what does the length dependent catastrophe mechanism need to be to achieve that steady state lengths, and so on. So this sort of helped us navigate the highly complicated parameter space that we're in. And then also we were able to validate with fluorescence data. So I just want to show you just the matching of the data and the validation. The main thing I want you to take away is that in red, we have the target mean that, at least for the 2 rows, we have experimental data on. Blue is the emergent mean. And I'm plotting this for 2 different situations, when we have length-dependent catastrophe and when we don't. And the main point here is we can achieve a physiologically reasonable speed distribution with both of these mechanisms on. So maybe the cell could be using both of these mechanisms in different contexts. And the second thing I want you to take away is I've got this length distribution at the bottom row. We by and large match the desired target mean, but we do get very different length distributions. So you have a Gaussian-like distribution with this length-dependent catastrophe, and you have more exponential-like distribution without that mechanism. And both of these types of distributions of lengths have been seen previously. Okay, well, we used that kind of data to help parameterize our stochastic model. Can we use other data to maybe validate? And so there's another type of data set called fluorescent tubulin turnover. And what that means is that you can fluoresce a certain part of the axon or dendrite. The tubulin within that domain region is going to fluoresce red. It's going to fluoresce red if it's contained in the microtubules. And once that tubulin pops off in shrinking, it's no longer going to fluoresce. And so what I didn't tell you, right, those red regions from before, that is that exact experiment. So we're able to, it's in silico, run these types of turnover assays. And what I want you to notice is that within 6 hours, you have complete turnover. So meaning the tubulin that was previously in that microtubule, after 6 hours is no longer the same tubulin. So these are highly dynamic. There's high turnover of these filaments. And so here what I'm showing you is these fluorescence intensity curves with and without those 2 mechanisms turned on. And here I'm varying the amount of protein available for these filaments to grow with. And so you see with length-dependent catastrophe, you have tight regulation. With no length-dependent catastrophe, the fluorescence intensity can vary quite greatly. But the wonderful thing about this is that this model result can actually inspire some hypotheses that can be tested in experiments. So right now in the Rolls lab, they're trying to make a genetic mutation of the cell to upregulate or downregulate tubulin, where you run these these experiments with these two different types of cells. And if you have a lot of tubulin, what does the fluorescence look like compared to a normal healthy cell? But these are all the results with fixed number of microtubules. What's known in this system is that nucleation is tightly regulated. It actually occurs in these branch points. And so, well, what happens if you vary the number of microtubules in this model that I just presented? So now we still have the same all plus end on one side, but rather than when a microtubule dies, I'm not going to automatically reseed it. I'm going to have some Poisson process that's going to allow more microtubules to join into the domain. And so we can do just that. So we can add this mechanism into this framework that we've previously developed. And we can ask, well, okay, well, how does the length distribution change as we vary this parameter? How does the average number of microtubules that are present in the domain, how is that its relationship with average microtubule lengths? And so I'll briefly share just two results from this. And they're honestly not terribly surprising. The first thing, we did do a similar continuous framework idea where we wrote down a PDE that describes this behavior. I'm not going to show that for time because I want to get to the extreme part of this, but come talk to me later if you're curious. The main idea is in red, we have high nucleation. We have a lot of very small microtubules, right? But with low nucleation, you have this more Gaussian-like distribution where you still have some somewhat long microtubules with low nucleation. And then the interesting thing about this framework, you can ask, well, what's the relationship, right? If you've got not many microtubules, what do their lengths look like? If you have a lot of microtubules, are they all really, really short? And so for high nucleation, you're pushed on this boundary of infeasible lengths given the amount of tubulin that you have available. And so with high nucleation, you have high variance in the number of microtubules, but low variance in the length. But with low nucleation, you have low variance in the number of microtubules, but incredibly high variation in the lengths of microtubules seen in the domain. So meaning, right, this is another result that could be potentially validated with these turnover results. And what I mean by that, it's much easier to change the nucleation level. If you have a high variance in the number of lengths, right, and you also have longer microtubules, you can imagine the fluorescence is going to be more persistent. And so you can really kind of tease apart, with these different nucleation levels, you can maybe potentially see that type of variance in experiments. Okay, so these are two models that we developed, but really the idea was to use this with the spatial component. Like, how do we incorporate these models of microtubule behaviors with this extreme behavior? Okay, and so the spatial mechanisms, there's three that the lab thinks are important. Number one is that you have this biased nucleation. So meaning if a microtubule appears at this branch point, it's going to grow in the direction of what's in that junction. You have what's called a checkpoint mechanism. So imagine you've got kind of like a QC, like I'm going to allow this microtubule to grow past the branch point based on some checkpoint procedure. And then the third mechanism is this idea, if you have a microtubule growing into the trunk, you have a kinesin motor that's going to dictate whether or not you steer up and away or down toward the cell body. And so for simplicity, let's do these two mechanisms. With these two mechanisms, can we get this maybe healthy condition where we have all minus end out microtubules in a certain domain? Okay, so and just to give you an example of, we have data on these mechanisms. On the left, I'm showing you a plot of the plus end out comets coming toward the cell body. So this is just telling you there's a lot of minus end out content in that part of the cell. And then the nucleation is a lot of minus end out. When we do this genetic knockout or we have an unhealthy cell, we have a lot of mixed microtubule polarity. The nucleation is also going to be mixed. So essentially the polarity is going to match, or the nucleation is going to match the pre-existing polarity at that junction. Okay, so all right, so now we can take our model, this nucleation model, we can nucleate a microtubule some rate nu, but now we have this additional probability that, okay, well, where is it going to be oriented? So we're adding this additional regulation into this process. Okay, so that's an example. We also implemented the checkpoint mechanism. This is all work done with recently graduated PhD student, Dr. Hannah Scanlon, who's going to Pfizer for a postdoc. But she took this comb dendrite and now is incorporating these two spatial mechanisms, where now we have that underlying microtubule model under the hood, but now we're also having this regulation at these branch points seen in sort of this peach color. And the question is, which one of these two mechanisms is really required to establish and maintain this biased polarity that we see in this dendrite, right? So I'm going to show you three different instances of running the model with 50% minus end out microtubules and 50% plus end out microtubules. And it's really amazing. So you'll notice very quickly that you get very different behavior. Moreover, right, sometimes the blue wins, sometimes the pink wins. Sometimes it wins quickly, sometimes it takes a while, right? So you'll see the left one, they're still battling it out for that complete, biased polarity, whereas the other two have reached their steady state. And so here, what I'm showing you two trajectories of the minus end out proportion within the whole domain, and you see that these trajectories rather quickly can achieve this extremely biased polarity. And then we do see that this polarity is maintained at an appropriate organism timescale. But what do we need? Like, which mechanism is in charge of this? And so this is showing you a histogram of the percentage of the minus end out microtubules, right? What we want is this bias, either all minus end out or nothing. So with neither mechanism, you get mixed. With just checkpoint, you have this very uniform distribution. But you see when we turn feedback on only, you get this biased behavior in the microtubule orientation. And so really, this is the mechanism that's sufficient to establish this biased polarity seen in this domain. Okay. So, but what happens in the extreme case? What's going on here, right? Well, here I'm going to show a little more movies from the PhD student in the lab that's creating all this data. So here we're looking at the pink region of this cell. All of these plus ends are going toward the cell body. When you sever the axon, very different types of behavior, right? You see a lot more, you see more, right? Because nucleation is upregulated. But you also see more microtubules that are moving away, or plus ends moving away from the cell body. And also, right, they're successfully leaving the cell body, whereas in the healthy instance, you don't see that movement up and away. And so what this meant for us is that, okay, well, let's take this spatial model and let's add this new boundary nucleation at the cell body, where we're going to allow plus end microtubules to enter the domain from the bottom at some rate. So here, but we're going to start with an all minus end out domain, and let's see, do we get the reversal? Does it happen? So it's going to happen at 2 hours, right? The minus end out polarity is maintained. And then at hour 2, we start inputting this plus end out microtubule at some rate. And you see rather quickly, you get this very large, global change in the polarity state in this domain. And this is all preliminary. Here, I'm showing you a plot that Hannah made of varying that rate at which we're seeding these plus end out microtubules in from the cell body. At 0, we maintain the minus end out rather well, but very quickly, if you have any type of input from the cell body, you can reverse this polarity, and rather quickly, right? You can see this very steep drop, even as this parameter just very small increases. So yeah, right now we're trying to parameterize the data with some preliminary data. We can track the quantity of plus end out microtubules in different regions away from the cell body. So this is really wonderful collaboration between biologists and mathematicians that are saying, hey, we need more data. We think this data would be good to parameterize our model. Can you get this data? And she's like, we absolutely can. And so there's some current hypotheses of what's going on in this interstate, where there might be a kinesin motor that's a positive regulator that's allowing these plus end microtubules to leave the cell body and enter the dendrite. So just to sum up quickly, I'll put this extreme video here, right? We have this minimal model. We have this wonderful iterative collaboration with biologists trying to understand this massive rearrangement. And hopefully this framework can be used to test other hypotheses that the lab comes up as to why we see this behavior. And we're working on adding even more complex geometry to this. But with that, I'll thank my collaborators, my funding, and I'll take some questions. Thank you.
questions for Anna?
Yeah.
Sort of like shaped would you get if you just let these stick grids like, and I think it was like what you touched on right at the end about the complex geometry. What sort of exploratory properties would we get with this sort of branching structure?
Yeah, that's a great question. I mean, I think that the idea, right, if you injure the axon, you want to reestablish the connection to the downstream neuron, right? So yeah, you're going to have a dendrite that's going to be searching for that cell. And I mean, I can imagine it can be pretty varied. I think there's a video, I mean, even this video here, right? I mean, it's, I think it can get pretty complicated. But I haven't looked at all of the images that the labs produce, but this is a pretty well-documented phenomena. So I'm sure they have data of what kind of paths they take, but I don't know off the top of my head.
I remember reading a while back about how preceding these catastrophe events, there's actually this like finite, sorry,
No.
this like finite period of just kind of this weird
Like a
instability.
pause.
A
Yeah.
pause.
Stuttering, right?
Yeah.
Stuttering. Thank you. Yes. I was trying to learn this because I was
Yeah.
trying to talk about it. Anyway, like you have this stuttering. Is that related to this like polarity business? Like I feel like people at least 2 years ago or 1 year ago didn't really understand why stuttering was happening. They just saw it and knew that it could be like a predictor of catastrophe events.
So yeah, yes, that's a great point. Stuttering, pausing, there's a lot of different states that the ends can be in.
Right.
In terms of polarity, I don't know. The second thing I'll say is we're really trying to take the framework, the mindset of what is the data that we have. And because the in vivo data is so coarse grained, right? These microtubules, this is not in vitro experiments. And we're keeping that in mind. We don't want too many parameters that we can't really quantify without asking, Melissa to really look at closely what's going on. So I would imagine that things are happening that related to stuttering, but
about how the experiments work, but I wonder if looking at polarity could tell us about when stuttering happens and when it doesn't, for how long.
yeah.
Like the spatial constrictions of
Yeah, that's true. That's interesting. Yeah. I hadn't thought about that. Barkov, yeah.
Is there like a spatial correlation on like the new dendrite that's like chosen to be?
Yeah, that's a great question, right? How does it get chosen? Who gets picked to be the new axon? Right, when they discovered this event, they had no idea. Now, data suggests that the more simple the dendrite is, so the topology is not complicated, that one is more likely to get chosen, but it might be related to the branch angle from the axon. We don't know. That's another, so by incorporating this sort of comb-like geometry, we're trying to get at why is it that the simple, morphology is getting chosen over this complicated comb? But yeah, they don't know.
Okay, let's thank Anna again.